Okay, well, good afternoon, everybody. I'm going to sort of slightly take a little bit of a different tack in the sense that one of the principal purposes of us gathering here today is to figure out how we can actually establish new partnerships. And I've been involved in a number of these type of different events, so I'm going to sort of give you a little bit sort of background of what I'm kind of interested in, and then how I have come to establish different types of partnerships with different faculty members across different colleges. It's great that we have all these activities that are really well developed, and these are highly successful collaborations. But the question is how do we continue to build those and develop those? And so I'm going to talk about sort of examples from my own personal perspective on how we have been able to do that in a smaller way, which will be leading to larger types of things. So what I'm going to be talking about in general is going to be about electronics, the use of electronics, and plants. But I'm going to digress a little bit to begin with, because basically it's all about communication, not just in terms of what I'm interested in, but what we're all interested in terms of how do we establish new partnerships, and it's through different types of interactions and how we figure out how to facilitate these type of interactions to make things work. We can't always jump in at the deep end, so to speak. So I'm going to talk about communication as it relates to plants, specifically how plants can protect themselves from pests and pathogens. And there are various types of signaling systems, communication pathways that are involved in that. And I'm going to sort of walk you through a little bit of my background, how I got to where I am now. And the question is that there are these signaling systems. Plants don't talk, but they have a language. So how can we eavesdrop or extract this type of information about how plants are communicating, and how can we use that information for, in this instance, to improve agricultural productivity? And as you'll see, that this sort of highlights the need to court these different types of interactions between different groups of expertise, whether it be me with plant background or other people be engineers or with chemists who have different skill sets that we can bring together, come together in different scales to be able to develop solutions to some of these underlying questions. Okay, so I'm going to go back to early days, for me anyway. So I was amazed when I was a student. I discovered that plants, like animals, have basically an inducible defense system. And this was quite revolutionary to me at the time. I mean, this is quite a while ago. And now we refer to this as acquired systemic resistance. And this is actually a diagram from some of my own work from years ago. But basically, what we're seeing here is that on the right-hand side of the plant, there's an induction, there's a small stimulus that results as we move around clockwise to the release of these systemic signals, which then can activate the plant so that the plant now becomes eventually protected from subsequent infection. So it's all about these mobile chemical signals that are moving throughout plants. And interestingly enough, the compounds that we helped discover involved in this are sort of related to aspirin. It's not actually aspirin, it's salicylic acid and some other derivatives. And these have subsequently gone on with work that we did and others have done with a number of the ag biotech companies, have led to commercial products. And one of the most well-known is something called Actigard. So this is a chemical that it's not a fungicide, it activates the plant's own defense mechanisms. Amazing. Anyway, so from there, I moved on to work on another signaling system, and I worked on something called cyclic AMP. And this is a derivative of ATP, and cyclic AMP is one of the central signaling molecules in life. Not just in plants, in life. It's a second messenger. It relays communication from one sort of biochemical pathway to another and amplifies that. I mean, I don't really have a good analogy, but it's like the baton. The baton, to win the race, you have to get the baton around in a relay race. And it's the same kind of thing with these signaling molecules, these second messengers, is they have to relay this information across systems. And so we discovered quite some time ago, more than 20 years ago now, that this system is crucial to developing defense pathways in plants. And the way that this works is that it's a phosphorylation relay. So proteins get phosphorylated. As they become phosphorylated, they become active. So, yeah, we all think about genes as being the critical element in our bodies. They are, because they encode the proteins. But the activity, the actions that occur within our cells is due to changes in those proteins. And one of the critical changes is through phosphorylation. So this has led to this field of what we now think of as phosphoproteomics. And I know that Dave Muddyman is here in the back row. I see him sitting there. So it just happened that I was in a seminar for another student in my own department, and who Dave was actually working with, and suddenly mentioned that they have this expertise to do this. And all of a sudden, this sort of opened my eyes to the opportunities that we have to actually develop this new technology to understand plant defense mechanisms. And so I've worked with Dave for more than 10 years, and it's been a really interesting and successful partnerships. We've got more than 12 publications. There's more coming over the period of 10 years. And it resulted in fabulous funding opportunities. We were funded by the NSF, competitive grants from USDA. So again, this is an example of how that, just through a happenstance and the curiosity that one has, to explore these type of partnerships. So that brings me to now. In 2016, I took a sabbatical. And on that sabbatical, I worked on stuff completely unrelated to what I normally work on. But I really encourage everybody to think about a sabbatical, because it's the time you get to think about what it is you're interested in. And I've always been interested in communication. And so when I came back from my sabbatical, there was a workshop similar to this in many respects, that was organized by CALS and the College of Engineering. I think people like Becky Boston and Alan Rebar, and I forget a bunch of others, Michael Steer, John Gilligan, and a few others. And what we did, we had, and I'm sure a number of you have probably been involved in these things, are these quad charts. It's a single slide where you make a pitch. And it was, so we basically, I think it's probably going to be like this session that's coming up. So it's like you get like a minute to make your pitch. And it was remarkable that at this event, which I had no great aspirations of, I was energized, I was back from a sabbatical, I was ready to take on the world. And it was amazing. At this particular workshop, the number of opportunities that seem to exist between the plant scientists and engineers to work on sensor-related technologies. And I thought, wow, this is incredible. Why didn't I know this before? So it was a real eye-opener, was this type of opportunity. And I've listed a few things that came out of that. So there's developing sensors for volatile organic compounds, and I'm going to talk about that, but there was others of sort of on-plant sensors using nanocellulose, or measuring changes in metabolism through drone technology and other stuff. But it was remarkable that there was this underlying commonality across our two colleges that it took me, I've been here for 20 years, to figure out that there was this common area that we could work together on. Anyway, so that really changed my outlook. And so let me tell you a little bit about plant volatiles. So we all know plants can smell nice. We all know that plants can not smell so nice. We all can detect a moldy basement, okay? So it's clearly that volatiles are playing a very big role in everything related to plants and microbes and other things that are going on in the environment. And we know a lot about the pathways. These pathways, these core metabolic pathways, I mean, you can see some of them here. I mean, there's things like isoprenes and phenylpropanoids and other stuff that's on here. You've got, I can't read this very well, but sesquiterpenoids and hemiterpenoids, and you've got jasminates. And then you see up there the green on the top right, the green leaf volatiles up here somewhere. Again, we all can smell, we all know when someone's cut the grass, right? Because they're releasing all these volatiles into the air. Why do plants do this? Good question. They are screaming. They are indeed screaming. And mainly they're screaming for help, okay? And so I sort of, here are some of the different things that go on when plants are releasing these volatiles. Plants are doing it for a variety of reasons. They can warn other plants of impending danger, so that the neighboring plants can activate their own defense mechanism. They can attract pollinators. They can attract animals to disperse seeds. They can attract predators that can come in and attack herbivores. So there's a lot of different things that all these volatiles are doing. They can activate defenses, attract beneficial insects, warn adjacent plants. An amazing world of the plant volatiles. We can think of this as plant talk. How the plants are communicating, they have their own specific language. And this language is composed of more than 1,000, potentially 2,000 different volatile compounds that are released from plants, okay? Not every plant produces every one, and I showed you the main pathways, but it's those tips, it's the leaves, okay? And that's where the specificity comes in. And indeed, it's the composition of these particular volatile profile, okay, that is a sort of a diagnostic signature of what's happening in a plant. So how can we detect and exploit this? And so this is where these informal discussion leads to this partnership. And this partnership, there's myself, Sophia Carthuriou from food, bioprocessing and nutrition. She's interested in foodborne illness and how can we detect foodborne pathogens. Greg, who did the introductions, interested in how can we modify materials as detectors in life sciences. And critical to this is Omar Oralkhan, whose name you saw on the previous slide. So we had this great idea about developing these sensors, and we said, well, how can we get this off the ground? So we put together a small proposal. We took it around our different departments, our different colleges, and boom, we found someone who was willing to bite. And this was the North Carolina Agriculture and Life Sciences Research Foundation. Their purpose is to raise money, not to spend money, it's to raise money. But we made a good pitch to them. And we were able to show that we could gather additional resources from our home departments to put together and leverage and make a package. And they bought it, they bought in. So the foundation of this is this technology. And it was introduced a moment ago, is this particular type of sensor. So it's a gas-based sensor, and as we heard, it can have different channels, so the channels can be switched out. And it's low power in this instance. It's driven from a watch battery. It's about the size of a quarter. It's wireless, it's low power, et cetera, et cetera. And this was, as we heard, is developed as a wearable device. So the question, so how does this thing work? Well, basically you can think of this thing as a little micro drum. So on the left, you see this little symbol there, and what happens is if you put a functionalized layer on this, and you change the properties of that layer, it will bind different molecules, okay? So when molecules bind, it changes the resonance frequency. It's a little micro drum, and so the resonance frequency changes. As you can see there in the middle panel, when molecules interact, changes the frequency, okay? When you remove the molecules, it goes back to baseline. So what we can do, and this is what we're working on, is you can modify the different functional properties, okay, of these different surfaces. So you can see that in this example, we have 6 different functionalized layers, and you can see that they will respond to different volatiles in a differential way. So essentially, you get a unique fingerprint. So that's the idea, to do this. So does it work? Can we do it for plants? Can we use it for plants? Well, this is our first experiment, and bingo, a huge response. So in this, you see this corn plant inside this glass dome with a sensor inside of it, and we wound the plant. And you can see that essentially immediately upon wounding the plant, there's a huge release of volatiles, and we can detect them. Fantastic. So we've gone on, now we've built multi-sensor arrays. So we're trying to develop different sensors that have different sensitivities, different properties, and we can see here that upon wounding, again, we see differential responses. We haven't yet demonstrated that we can actually distinguish the different types of molecules, but that's where we're going with this. Not only plants give off volatiles, fungi, the moldy basement. This is really important because many fungal pathogens produce a variety of noxious compounds, and many of them are volatile. So here's an example where we don't necessarily have to detect the disease. We can detect that signature of the infection. Okay, we need to identify what the compounds are, so we do the standard approach. We can see some nice red peaks here. So these are different compounds that are released during that few minutes, different than we see in a later time. Okay, so we can check these, so that's what we're doing. When we have the presence of a particular volatile, the sensor goes off, etc. So we're now distilling down, figuring out what these different volatiles do. The other important thing about this was that we wanted to generate some real excitement about the possibilities of using these type of technologies. So myself, in collaboration with Omar, we approached our college, the College of Agriculture and Life Sciences, and in collaboration with the College of Engineering, we co-organized and hosted a major conference, and this is part of the Stewards of the Future conference. And this took quite some effort to make this happen last fall, but the Stewards of the Future series is one of the flagship symposia that the College of Agriculture operates. And this was really a very impressive event. We were able to generate a lot of resources to come into this to support this particular thing, this particular activity. We had worldwide speakers and we had worldwide participation. We had 150 people paid good money to come to this thing. So it really enabled us to get this whole activity to the next level. So let me just conclude. In a short period of time, through perseverance and having, I think, what was a really good idea, we've got a productive and a visible partnership between faculty and our two colleges. We're developing a technology. This technology was developed here at NC State, which is absolutely wonderful. There are endless opportunities and applications for this type of technology for basically sniffing out, sensing different volatile compounds. We've been successful in getting corporate sponsorship. We have a major sponsor and recently got an NIH postdoctoral award to support this, so it's all moving in the right direction. And I should say that this is just part of a much larger initiative ongoing here at this university. There are other fruitful collaborations between members of CALS and engineering. And Kranos Williams, I know he's here today. I think he's going to be presenting or discussing some things about some other global opportunities for partnerships relate to this initiative. So overall, in a very short period of time, the future looks very bright. All right, that's all I have to say. Thank you.
So that the folks listening online would be able to hear the question in addition to your answer, that would be great.
Okay. Yeah.
What are the VOCs doing when you wound the plant? I mean, are they doing something to help the plant heal?
So the question is, what are these volatiles doing when we wound a plant? So you're releasing a lot of terpenoids and green leafy volatiles, which typically we can smell. But it's very interesting that they're actually warning neighboring plants. They're warning a neighboring plant that we're potentially under attack from a herbivore. Activate your defense mechanisms. So there's well-established literature on this that these volatiles can travel many meters and they can activate some of the classical jasmonate and salicylic acid pathways in plants, so the plants can be ready. And it all ties back to some of my original work on systemic acquired resistance. It's the same general idea that these are mobile chemical signals that are involved in activating plant defense mechanisms. You're welcome.